Hello, and welcome back to the Prentice Hall Biology textbook. Today we'll be covering Chapter 10, Cell Growth and Division. Okay, 10-1, Cell Growth. So most things grow by producing more cells. The cells themselves don't actually grow, there are just more of them. And there are two main reasons cells divide rather than grow indefinitely. So the first one is the DNA overload. As cells increase in size, they don't make extra copies of DNA, so there are more organelles needing genetic information from less DNA. The next is the exchanging of materials. So the exchange rate of food, water, and waste depends on the surface area of the cell. And the more surface area, you have more surface area with smaller cells and less with smaller, more cells and less surface area with fewer larger cells. So the rate of production then is dependent, the rate of production of waste and usage of food is dependent on the volume of the cell. And understanding the relationship between a cell's volume and its surface area is key to understanding why cells must divide as they grow. So this brings us to the surface area to the ratio of the surface area to volume. So as the size of a cell increases, the ratio of the surface area to volume decreases. And it's better for cells to have higher ratios. It makes them more efficient. So if it gets too large, it can be difficult for it to get sufficient amounts of oxygen and nutrients in and waste products out. So here on the screen we see a 1 millimeter cube has a surface area of 6 millimeters squared and a volume of 1 millimeter cubed. So that's a 6-to-1 ratio. But as we increase the size to the 2 millimeter cube, we see the ratio goes down 3-to-1, and to the 4 millimeter cube, it goes down to 1.5-to-1. So if you had 64 1 millimeter cubes, which is the same volume as one 4 millimeter cube, you would have a much greater amount of surface area. Okay, and then division of the cell. So the cells, before they become too large, must divide and form two daughter cells. And each of these daughter cells is an identical version of the original cell. And before division, the cells must replicate all of its DNA so that each daughter cell can get its own genetic information. And this increases the surface area to volume ratio. Okay, 10-2, cell division. So there are two main parts of cell division. There's mitosis, which is the division of the cell nucleus, and cytokinesis, which is the division of cytoplasm. So reproduction by mitosis is classified as asexual. That means there's no sexual reproduction. It turns from one cell into two cells. And this is done in most, if not all, eukaryotic cells and prokaryotic cells. So in eukaryotic cells, the genetic information is passed on through the chromosomes, which is condensed chromatin found in the nucleus. And now humans have 46 chromosomes, and we do not see the chromosomes until the start of cell division. So before cell division, each chromosome is replicated because each chromosome consists of two identical sister chromatids. So one chromatid goes to each of the new cells. We'll get into this more a little bit later when we talk about mitosis. So the cell cycle. There's interphase, which is the period of growth in between cell divisions. So the cell cycle is a series of events that cells go through as they grow and divide. And during the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells, in each of which then the cycle begins again. So it consists of 4 phases. There's M phase, which is mitosis and cytokinesis, S phase, which is chromosome replication, and then G1 and G2 phase, which are gap phases, and during these phases there's periods of intense growth and activity. So the events of the cell cycle. So interphase is divided up into 3 phases: gap 1, S phase, and G2. So gap 1, the cells do most of their growing, synthesize new proteins and organelles, and then during the S phase, chromosomes are replicated and key proteins synthesized. And in G2, the organelles required for cell division are produced. So over here we see it starts at G0, and then it's G1, that's the growth phase, and then we have S phase, which is DNA replication, and then G2, which is the production of organelles, and then it goes into M phase. So, or into mitosis. So mitosis is divided up into 4 main phases. We can see them here. There's prophase, metaphase, anaphase, and telophase. And here is prophase. So prophase is the first and longest phase. During prophase, the chromosomes become visible. The chromatin is condensed down into chromosomes, and the centrioles, the two tiny structures located in the cytoplasm, are they separate and go on opposite sides of the nuclear envelope. So the centrioles lie in a region called the centrosome that helps to organize the spindle, which is a fan-like microtubular structure that helps separate the cells. The condensed chromosomes become attached to the fibers in the spindle, and we see the nucleolus disappear and the nuclear envelope start to break down. Next we have metaphase. So metaphase lasts only for a few minutes. Here we see the chromosomes line up across the center of the cell, and the microtubulars connect to the centromere of each chromosome to the two poles of the spindle. So the centromeres are what connect, they're these orange dots, they're what connect the sister chromatids together. Okay, so that's metaphase. Next we have anaphase. So anaphase, we see the centromeres split. The sister chromatins are separated and become individual chromosomes, and they move towards opposite ends until near the poles of the spindle, and then anaphase stops when the chromosomes stop moving. Okay, now we have telophase. So telophase is the fourth and final phase of mitosis. The chromosomes begin to disperse back into tangles of dense genetic material. The nuclear envelope reforms around each cluster of chromosomes, and the spindle breaks apart, and the nucleoluses become visible in each daughter nucleus. So with this, mitosis is complete, but cell division is not. Now we have two separate nuclei in one cell. So that's when we go to cytokinesis. So with cytokinesis, we have the two separate nuclei in one cell, and cytokinesis will split apart the cell membrane. So this occurs at around the same time as telophase, and in most animal cells, we see the pinching of the cell membrane until it pinches off and forms two separate cells. However, in plants, there's a structure known as the cell plate, which forms midway between the divided nuclei, and it gradually develops into a cell membrane and then a cell wall. Okay, 10-3, regulating the cell cycle. So cell growth and cell growth in the cell cycle are highly regulated throughout the body. With the controls on cell division are very precise. Cells, when they come in contact with other cells, will stop dividing so they don't overcrowd and produce too many cells. So some cell cycle regulators. So scientists looked for a substance that would tell the cells when to divide, and this was discovered by Tim Hunt and Mark Kirschner, who discovered that cells in mitosis contained a protein that would, when introduced to other cells without the protein, would start mitosis. So they called this protein cyclin, and cyclin regulates the timing of the cell cycle in eukaryotic cells. Okay, so then there are internal regulators. So internal regulators are proteins that respond to events inside the cell. So for example, a regulator protein makes sure that a cell does not enter mitosis until all of its chromosomes have been replicated. And then there are external regulators. So that are proteins that respond to events outside of the cell. Most often they direct cells to speed up or slow down the cell cycle. So this is when cells come in contact with another cell, the external regulators will tell that cell to stop dividing. And so they help to prevent excessive cell growth and keep the tissues of the body from disrupting one another. And so when these regulators aren't working, we have what's known as uncontrolled cell growth, which is results in cancer. So cancer is defined as a disorder in which some of the body's own cells lose the ability to control cell growth. And so these cancer cells do not respond to any signals that regulate the growth of most cells, and often they mutate, and the mutated cells will keep on dividing. So they divide uncontrollably and form masses of cells called tumors. Okay, key concepts. So for the key concepts for Chapter 10, we have give 2 reasons why cells divide and don't grow indefinitely. Name the 2 main events of the cell cycle. Describe what happens during each of the 4 phases of mitosis. And then what chemicals regulate the cell cycle and how do they work? What happens when cells do not respond to the signals that normally regulate their growth? All right, that's it for chapter 10. I'll see you at chapter 11.